Hello my dear students, this is Dr. 𝗲𝗵𝗺𝗺, your biochemistry faculty. Well, in this video we are going to discuss, this is in continuation with my previous video where we discussed about the metabolism of purines by de novo and salvage pathway. So now we are going to discuss about the metabolism of pyrimidines. Now, these pyrimidines are N1 glycosides, very important. जैसे purines are N9 glycosides, pyrimidines are N1 glycosides, and this is the structure of a generalized structure of purines. Numbering starts always clockwise. So 1, 2, 3, 4, 5, 6. This will come from, the major chunk will come from aspartate. This will come from bicarbonate ions, and this is going to come from glutamine. So this is the source, and since N1, so N1, and here it will be attached with ribose sugar like this, and so on. Okay, so now the question is where the synthesis of pyrimidines is there and what is the precursor of pyrimidines. Again, if we talk about this, the synthesis takes place in the cytoplasm. And here there is a similarity between this and urea cycle, which I have discussed in detail in the in my video on the urea cycle also. So here what happens is these bicarbonate ions combine with glutamine. Very important, these bicarbonate ions first of all will combine with glutamine to form carbamoyl phosphate, right, and here the enzyme is CPS II, which stands for carbamoyl phosphate synthetase 2, right, and obviously synthetase is there, so we have to write ATP, ADP plus inorganic phosphate. Now once this carbamoyl phosphate is produced, it will then combine with aspartate, because you see glutamine has come, this has come, now aspartate has to come. So the second step is that this carbamoyl phosphate will combine with aspartate and it is going to form carbamoyl aspartate, yeah, carbamoyl aspartate. And if we talk about the enzyme is aspartate transcarbamylase. Carbamylase, right. And once this carbamoyl aspartate is produced, then it is going to form dihydroorotate. Dihydroorotate, right. The name of the enzyme here is dihydroorotase. So these three enzymes represents a multifunctional enzyme, and to remember this is CAD. I mean CAD is the mnemonic, and this is a multifunctional enzyme. Or it is known as, we can say, CAD. Now once this dihydroorotate is produced, then this dihydroorotate will simply undergo dehydrogenation, which is the only step of pyrimidine biosynthesis occurring in the mitochondria. So I can say that because the enzyme is dihydroorotate dehydrogenase, so NAD will be there. And the enzyme is dihydroorotate dehydrogenase. Dehydrogenase, very important, right? And it is going to form orotate. Now once orotate is produced, automatically you can say this will undergo phosphoribosyl transferase forming OMP, and same reaction PRPP PPI, and the enzyme is orotate phosphoribosyl transferase. And this OMP will lose a molecule of carbon dioxide undergoing decarboxylation. So we can say the enzyme is orotate decarboxylase. And this OMP is going to form the first pyrimidine, which is uridine monophosphate. So the first pyrimidine which is produced is uridine monophosphate. Now this uridine monophosphate will undergo reactions to form the other two bases, pyrimidine bases, which are thymine monophosphate and cytidine, or you can say cytosine monophosphate. So what will happen here is this UMP will simply undergo kinase reaction first of all. So the enzyme is UMP, obviously kinase, and the donor is ATP. And it is going to form UDP, uridine diphosphate. And this uridine diphosphate will undergo reduction. Why? Because uracil is the base present in RNA. And RNA has ribose sugar. And we have to form a base of DNA now. And DNA, the basic difference is DNA has deoxyribose sugar. So this has to be converted into deoxynucleotide. So this UDP will undergo a reduction in the presence of thioredoxin reductase. Very important. Reduced will be converted into oxidized thioredoxin, and this will be converted into DUDP. And this then will be converted into thymine monophosphate. How? By taking a methyl group from folic acid. Okay. So what happens here is, this is a very important reaction. N5N10 methylene THF will donate its methyl group here, and thymine monophosphate will be produced, and the name of the enzyme is 𝗖𝗵PH synthetase. 𝗖𝗵PH synthetase. And if we talk about UDP again, this UDP will undergo one more kinase reaction, forming UTP, taking glutamine to be converted into CMP or CTP, we can say CTP, cytidine triphosphate. So this is how the synthesis occurs, and the most interesting thing if we talk about is this uracil. Actually this uracil is having this type of structure, 2,4-dioxo, like this. This is uracil. And this uracil, how it is converted into thymine? This is the structure of thymine. Double bond O, double bond O, CH3. So it's like 1, 2, 3, 4, 5, 6. So the structure of thymine is 2,4-dioxo 5-methyl pyrimidine. This is thymine. And from where this methyl group is coming? It is coming from folic acid. And this is the main feature of this, that majority of the anti-cancerous drugs are folate reductase inhibitors. Anti-cancerous drugs are folate inhibitors or folate reductase inhibitors. Very important. Right? And if we talk about the anti-cancerous drugs, for example, we can say methotrexate is there. So it is not going to let DNA to form, and that is what, because whenever a kind of virus invasion is there, they try to take the host machinery. So they have to be converted into DNA. But the reaction which is stopping the formation of DNA automatically will lead to inhibition of this. So how they are inhibiting? By inhibiting folate reductase. Clear? So this is what is the formation of pyrimidines. So we start with, first of all, you can see that bicarbonate ions combined with glutamine forming carbamoyl phosphate in the presence of CPS II. And this CPS II is the rate limiting enzyme. Very important. This is the rate limiting enzyme of pyrimidine biosynthesis. And this is present, obviously you can see it is present in the cytoplasm. Then it forms carbamoyl aspartate because the major chunk is coming from aspartate. Then ring closure occurs forming dihydroorotate, then orotate is produced, then OMP is produced, orotidine monophosphate in the presence of phosphoribosyltransferase transferase, and then this OMP forms UMP. In the end, this thymine is produced. Very, very important. So this is how it is synthesized. Then we come over to the most important part of this. What is the regulation? Now always remember, whenever we talk about the regulation, CPS II is the rate limiting enzyme. There are basically the CPS, it exists in two forms. It stands for carbamoyl phosphate synthetase. Carbamoyl phosphate synthetase. So this CPS exists in two forms. One is CPS I, another is CPS II. Now what is the basic difference between CPS I and II? CPS I is always present in the mitochondria, whereas CPS II is always present in the cytoplasm. Second is, this is a rate limiting enzyme of urea cycle, and this is a rate limiting enzyme of pyrimidine biosynthesis. Very important. And if we talk about the substrates in CPS I, because they are going to form urea. So the substrates here are bicarbonate ions and ammonium ions. On the other hand, here the substrates are bicarbonate ions and glutamine. Right. So this is how the synthesis of pyrimidines takes place. Very, very important. In the cytoplasm, there is only one reaction which takes place in the mitochondria. And this is anabolic. So then we come over to salvage pathway. Salvage pathway is also operative. Same, everything is same like we have studied in purines. De novo is there, salvage is there, everything is like that. Then we come over to catabolism. Now if we talk about the catabolism of pyrimidines, they always form two products. One is BAB, beta amino isobutyrate, and beta alanine. Right. So, beta alanine and this is no doubt increased in leukemia, but beta alanine is reused by the body. So, we can say that the catabolic products of this are reutilized within the body again and again. Very important. Okay. So, they are basically, you can say that the catabolic products of this are harmless. Just say if we talk about purines mein the catabolic product was uric acid, and we studied that hyperuricemia leads to gouty arthritis. Here none of that thing is there. Clear? Then after this, then we come over to what are the inborn errors of metabolism, or what are the metabolic errors or metabolic disorders. Now, metabolic disorders, the most common here is orotic aciduria. Orotic aciduria. So, basically what happens in orotic acid, urea, there is increased concentration and increased excretion of orotic acid. Now, if we talk about the reactions, we know that this orotic acid or orotate undergoes the reaction 𝗲𝗵𝗵𝗺 transferase and forms orotate monophosphate. And this orotate monophosphate then undergoes decarboxylation. So, I will just write here it is decarboxylation, or this is orotidine monophosphate. Ribosyl transferase. Right, so we can simply call it as OPRT also. The other name can be OPRT. Orotate phosphoribosyl transferase, and this is decarboxylase, and simply it is forming UMP. Now the question is, this enzyme can be absent and this enzyme can be absent. Whatever is the reason, there is increased concentration of orotic acid which is excreted in urine. So, it is known as orotic aciduria, where there is deficiency of either OPRT enzyme or there is deficiency of OMP decarboxylase enzyme. Deficiency. Then second here is Rees syndrome. Now in Rees syndrome, what happens is, now if I take you back to the first reaction is of urea cycle. If we talk about, in urea cycle, what is happening, bicarbonate ions combines with ammonium ions to form carbamoyl phosphate. This carbamoyl phosphate is combining with ornithine, forming citrulline. So, what is happening is, these reactions are taking place in the mitochondria. Now, what will happen is, here the enzyme is OTC and here the enzyme is CPS1. So, if OTC is deficient, there will be increased concentration of carbamoyl phosphate. Okay. And if we talk about the cytoplasm mein parallel reaction is going on. In the cytoplasm, bicarbonate ions are combining with glutamine to form again carbamoyl phosphate, right, which forms orotic acid and uracil, like this. So what will happen is, if there is increased production of this carbamoyl phosphate, it will be diverted here. It will enter into the cytoplasm. So you can say that it is going to increase the concentration of uridine monophosphate, concentration of orotic acid. So whenever there is deficiency of OTC, always remember OTC deficiency always leads to increased concentration of orotic acid and increased concentration of uracil in urine. And that is the basic difference between hyperammonemia type 1 and hyperammonemia type 2. What is the basic difference? Hyperammonemia type 1 means the deficient enzyme is CPS1 and here the deficient enzyme is OTC. And OTC is X-linked. So I will just write it here because we have discussed this in detail there also. So hyperammonemia, if a patient is complaining of type 1, there is increased concentration of ammonia, you can say encephalopathy is there, you can say respiratory alkalosis is there, that means the concentration of ammonia is more there. So what will happen? What will be the difference? The difference here would be 1 and 2. So in type 1 and type 2, first of all, if we talk about the deficient enzyme, so deficient enzyme here in type 1 is CPS1 and here deficient enzyme is OTC, ornithine transcarbamylase. And if we talk about the biochemical manifestations, here there is increased concentration of plasma glutamine, right. Here also there is increased concentration of plasma glutamine. But if we talk about uracil, here no orotic acid is there in urine, orotate in urine and no uracil. And in type 2, obviously orotic acid, orotate will be excreted in urine. So here obviously if we talk about the treatment, this patient will have simple, you can say uridine will be given as a treatment, right. So this is difference between type 1, type 2 and this is the synthesis of pyrimidines by you can say de novo pathway or salvage pathway. So that's all in this video. I hope it is clear to all of you. Thank you so much.